is gone. So whatever the mitochondria in the egg, it is from that's what either the son or daughter, all the mitochondria is derived from mother. Okay, if the DNA is defective in the mitochondria or in the nuclear genome, obviously people end with disease. Okay, so this is the mitochondrial staining, this is the nuclear genome, and then this is the green color is the mitochondria. Okay, we can able to differentiate the nuclear genome and mitochondrial genome. Okay, so we are for the past 25 years, we are working on many eye diseases. Some of the disease, see, this is the classification of the disease. One is the chromosomal disorder, single gene disorder, and then complex disease. Please recall your family, your grandfather or your uncle, they may have some disease. I am not talking about the infectious disease. Infectious disease in the sense, it will be like a COVID and other thing. No, I'm the organism. You are microbiology people are here. Like a bacteria, fungus, virus. It causes the disease. So I am not talking about that one. The disease related to the gene. If the gene is defective, they will end with this kind of disease. This is the disease. These are the name of the disease. If you come to the chromosomal disorder, only is there any addition and deletion. What is the monopoly, we call it body party like that. No. So is there any problem there? They have this kind of disease. But the percentage, you can see the percentage, the occurrence is only in 0.18 decimal. But in the chromosomal disorder, single gene disorder, single disease, I told you 19,000 genes are there in the human, 547 genes for eye disorder. If even one gene is defective, they will end up with this kind of disease, color blindness, cystic fibrosis, like that, hemophilia. I think for girls, please eat well and then you check your hemoglobin constantly. The Indian woman, the hemoglobin is only 8. You have to be 15 or 20, more than 15. Please check your hemoglobin content and then lot of what to say that dates. Okay, line dates. But they added some sugar, that is a different story. Better to consume some iron content food to enrich your blood. Okay, so these are the single gene disorders, and you see the percentage again is 0.6%. And come to the complex disease is 50%. This is the very late onset. So here, I think I mentioned in many places, the population is increasing like many times. So the elderly population is very high, more than 50 years people are living. The number is very high than the younger population. So that's what, they are prone to develop this kind of diseases, either the cataract or diabetic retinopathy, diabetes, glaucoma, like that. So cancer. So everything, I think there is a gene behind it. If the gene is mutated after the very late onset, they will end with this kind of disease. But again, you can recall, if there is any family history, your father, grandfather, if they have that kind of disease, you better to make your mind yourself, yes, I may turn myself this kind of disease after some time. So to avoid that one, I have to make comfortably, I have to lead a different kind of life and compared to my parents, to avoid or postpone the onset of the disease, those diseases. Okay, so I think this is the mode of inheritance. So it is autosomal dominant, autosomal recessive. If it is autosomal dominant in the sense, father is affected, and it is a kind of vertical transmission, like that. For grandfather, father, grandson, everyone is been having that disease. That is called the mode of inheritance is autosomal dominant. Okay, so if it is a recessive, your grandfather may have the disease, but if your son, the grandfather, son, grandson, the son may not have the disease, but the grandson may have the disease. It means the recessive disease, it will skip one generation. But you don't know what is going on, why it is suddenly appearing in the family. So it means definitely there is a family history because of the consanguineous marriage. If it is a relative marriage, if you marry uncle, the niece, that kind of problem, is there in recessive disease. And then the effective, this is the here, the severity of the disease is very low, but the severity of the disease is very high. That's what people are recommending. The relative marriage, don't do it. Okay, this is excellent recessive, excellent dominant, and this is one. I think wait, it's not okay. See, okay. See the mitochondria. Here you can see the mitochondria. So as I mentioned, the part of the mitochondria we have It is from mother. So if you see here, the mother, I think is having the mitochondria. He is having the mitochondrial mutation. All these children, is, having the problem. But okay, see here. This is affected and unaffected. Okay, so all are affected, and the father is not affected. So that's what they are unaffected. Okay, so this is, it means whatever the mitochondria we have got it from mother. Okay, so you can using the mitochondria. So that's what the secretary mentioned this kind of the information. So you can very long back they assess using the mitochondria how the human population migrated from Africa to different other continents. Okay, so what you can do? So you simply take the blood, isolate the DNA containing the mitochondria, and then you and me having the same kind of variation. That is, we belongs to southern part of India. We have the same kind of genetic marker that is called. Now we are all belongs to one particular haplogroup that is called M haplogroup. So we are belongs to this particular M haplogroup, and based on that, we can be able to track from how the human is migrated from different continents. That is possible. Okay, and then another catch is, if the mutation is there in the mitochondria from mother, obviously it is inherited to the son and daughter. The disease. There are some IDCs there. I will come to the point. Okay, so now I think because of the lack of time, I have to leave within 1 hour. So I think please, I'm requesting you give me another chance. I can interact with the students in a different way. So this is what I'm trying to say is microbiology. Suppose using the multiplex PCR, you can set up a PCR reaction, and then you throw all the primers. It is a bacteria, virus, a human, anything, it's a kind of multiplex. You put in one tube and then add the DNA, affected patients. Your target is multiplex, and then you want to identify only one particular disease if the patient is having. So it is amplifying only that particular fragment. There is a specific kind of reaction. So that is called, through this technique we can be able to identify. So whether he is having that particular bacterial disease or viral disease or a human mutation. Okay, so that is the story. And right now, so what we are doing in the lab, we are not focusing on one particular reaction. We are concentrating the entire genome. As I mentioned, 19,000 genes. We are taking the blood from the patient, and then we are focusing on what is the change in the patients, either in the 19,000 genes or only in the 540 genes. That is our target. Then only we can able to say, yes, you have this kind of a disease. So this is, we are adapting the whole exome sequencing and the whole genome sequencing. We have this kind of an instrument in our lab that is called MySeq and HiSeq. So using that one, you can be able to identify what kind of variation for the patients. Okay. So and then see here. So this is the patient. So this is the technology. So you can either use the genome or exome sequencing, or you can target with the transcriptome, or as I told you, the epigenetics. Epigenome. I told you. Even what is the histone modification? What is the CPGL change in the patient? So based on that, we can have the data analysis. So in the sense, here I think the bioinformatics system department is there. So once you get the raw data, so you can analyze the entire raw data and identify what is the specific variation in the patients after a particular gene. So we have our own pipeline. You take the data, put in the pipeline, and then you find out. So what is the cause of the patient? See what exactly changed in the associated with the disease. We can come to a conclusion. Then we can give the genetic counseling, or we can give for what is the mode of, how to change the mode of treatment. So that is possible. So I think this is the information for the bioinformatics group. I think if you have the large data, you have to carefully analyze which data is useful to the patients. Okay. Okay. So now I think I am working for the past 23 years on eye. You already studied in the 10th grade what is the structure of the eye. So this is the structure of the eye. So this is cornea, and then this is the lens, and then this is the iris, this is the ciliary body, and this is the optic nerve. This is the posterior chamber, this is the posterior chamber, this is the anterior chamber. Okay, so please, as I mentioned, gene is very important, right? So suppose, please come, switch the key to the developmental biology. So during the development, if